Hello, welcome to your online lecture for the course Pathophysiology 1. In today's lecture, we're covering genetic diseases. In particular, we'll be focusing on mutations, chromosomal aberrations or alterations, and then we'll finish up by talking about how genetic diseases are transmitted. Before we get into the pathophysiology of genetic diseases, we should review some normal anatomy. First of all, if this DNA that you can see here in its supercoiled form wants to replicate itself, it needs to unwind. And remember that DNA replication occurs during S phase of interphase during the cell cycle. We want to replicate our DNA so that we have equal DNA in either cell after mitosis. In order for DNA to replicate itself, though, it needs to unwind and unzip, which you can see starting to happen here. And you can see what it does is it exposes the nitrogenous bases. And DNA has nitrogenous bases cytosine, guanine, adenine, and thymine. And when these bases are exposed, it allows for its complementary match base to pair with it. And recall that cytosine and guanine will always match with each other. They are considered complementary base pairs, and adenine and thymine as well will always match with each other. And so what you can see happening with the help of DNA polymerase is we're going from one DNA strand to forming two DNA. So we end up with a replication of DNA. Now just a few minutes now on transcription and translation, which is different than DNA replication, which we just spoke about. So again, DNA will replicate itself during S phase of interphase in order to prepare for cell division. But what is the ultimate purpose of DNA? Well, we know that it carries the genetic information to produce a variety of different, so many different proteins in the body. But the first thing that has to happen in order to get ready to produce a protein and give the proper instructions is it has to be transcribed. So in the nucleus, what ends up happening is a particular segment of DNA, so we would call this then a gene, is going to be made into a copy, and that copy of that genetic information from that gene is going to be in the form of mRNA. I'd like to remind you that mRNA or RNA possesses the base uracil instead of thiamine. We have uracil. And what will end up happening is this copy of that segment of DNA called mRNA, once it's been edited and spliced and really finalized into its final form, it's going to leave the nucleus. And you can see that happening here, that it's leaving the nucleus and it's going to travel to a ribosome where protein synthesis will occur. I'd like to remind you of the term codon. A codon is a set of 3 nucleotides. So just for example, this could potentially represent a codon, this could represent a codon. And the ribosome is really going to be reading the RNA at 3 nucleotides, or rather 3 nitrogenous bases at a time. And that's called a codon. And a codon is then part of RNA. Once that mRNA reaches the ribosome, tRNA is now involved. And tRNA is going to be bringing a specific amino acid to the ribosome. And it's going to be a specific amino acid that's meant for a particular area of the mRNA. You can also see that tRNA possesses what's called an anticodon, and really it's the anticodon that will match perfectly with the codon at the level of the ribosome, and you can see that happening here. And then surely, one by one, as transfer RNA brings a different amino acid to the ribosome, we end up forming a whole bunch of amino acids that are bound by peptide bonds. And this forms our polypeptide, and then once we get other polypeptides together, we can form a protein. But the most important thing to take from this slide then is that in order for a polypeptide and then a protein to be formed properly, it's dependent on the sequence of amino acids to be in a particular sequence. And as soon as there's a change in the amino acid sequence, in other words, as soon as there's a change in what is actually originally coded for the development of that polypeptide or that protein, then problems can arise. And we'll talk about that on upcoming slides. We'll start with mutations. So a few important definitions that you need to know of with respect to the topic of genetics. The first one being mutations. A mutation is, as stated on the screen, a change in the sequence of a gene. And there are over 20,000 genes in the human genome. A gene, as I already told you on the last slide, is a segment of DNA, and it contains very specific information coding for a particular polypeptide and subsequently a protein. A mutation will involve any alteration of this genetic material or of this DNA. And this can be something that's inherited, but not necessarily. It doesn't have to be inherited. Mutations are considered very rare events in the grand scheme of things. Now, as a cell prepares to divide, remember this includes the replication of DNA, which happens during S phase, several checkpoints exist in the cell cycle where the cell or the DNA is going to be checked in order to make sure that it's normal. And let's say DNA is discovered to have replicated abnormally or something's abnormal within the cell. In a lot of cases, our body can take that cell, put it into a repair phase, repair any of the issues, repair the DNA, and then let that cell re-enter the cell cycle to continue its process for cell division. Sometimes, though, we can't repair these errors, and in these cases, our body will hopefully undergo the process of apoptosis, or programmed cell death, in order to not allow that cell to divide. Once we realize it's no good and we can't fix it, we don't want to be replicating it, because once you divide an unhealthy cell, it will just continue to divide at that point. And this is where conditions such as tumors and cancer, etc., can occur. A mutagen. This is an agent that's known to increase the frequency of mutation. Now, not all mutagens are considered equally potent, meaning that some are more likely to cause problems than others, and in lesser quantities potentially. But mutagens can be physical. An example is ionizing radiation, so an X-ray. They can be chemical. An example of a chemical could be formaldehyde and sodium nitrate, which have different potencies in terms of how mutagenic they are. And they can also be biological, such as certain viruses. For instance, HIV has the ability to dump its infected DNA into a healthy cell, infecting that healthy cell's DNA, causing mutations to occur. But we're exposed to mutagens all throughout our lifetime, and a lot of times they don't end up causing any problems, but of course they still can. Now, generally speaking, the effects of a mutation are commonly irrelevant to cell function and are therefore considered to be silent in a lot of cases. So it doesn't change the function of the cell. It's such a minor mutation, so it doesn't have any grand impact. And so these silent mutations are ones that can't really be detected under most clinical situations. So they're not very evident. Genetic testing may yield that there's been some mutation, but it's not otherwise obvious. In other cases, it could be sublethal. It could be something that then could lead to a disease. Lethal mutations can cause, for example, miscarriages or stillbirths. Two main types of mutations exist. We have base pair substitutions and frameshift mutations, and we'll speak about the specific types of each of these on upcoming slides. I'll let you know now that we will not be elaborating any more on silent base pair mutations because we've already really discussed them. They don't really have they're not obvious clinically. And the reason that they're not obvious clinically is because it doesn't really affect the sequence of a gene. Okay. So let's talk about, just actually go back here for a moment. For base pair substitutions, we're going to talk about missense and nonsense. Base pair substitutions exist as really three basic types, but two we're going to focus on here, and those are, as I said, missense and nonsense. Missense mutations occur when one base pair is substituted for another. One base pair is substituted for another. And this will cause a single amino acid change in most cases. And I'll show you what that means in a moment on the image. But because of the redundancy of our genetic code, sometimes there's no consequence of this, or there might not be any profound issues. So let's have a look at this image here. I'll get my pen so that I can circle the example. Here you can see we're going to talk first normal DNA. This is the normal mRNA copy of a gene, and then here would be the polypeptide formation. And remember that polypeptides are going to be amino acid attached to amino acid attached to amino acid by peptide bonds. In this normal DNA sequence, you can see that we have guanine and cytosine, and mRNA has taken a copy of guanine. And then we have this codon here, which consists of 3 nitrogenous bases, and 3 nitrogenous bases will code for a very specific amino acid. So their position in this order will code for a specific amino acid, meaning that tRNA, transfer RNA, is going to bring a particular amino acid, in this case this one, to the ribosome where it sees this codon. So it's attracted to this particular codon or group of 3 nitrogenous bases. But with base pair substitution, what you can see happening is that GC is now substituted for AT. So GC, that complementary bond gets kicked out and adenine and thymine move in. Nothing else, though, has changed if you compare both the normal DNA to this missense mutation, except for the fact that adenine and thymine as a base pair substituted themselves in. And so when mRNA makes a copy, you can see that it now takes adenine as part of it. And now here's our codon, which has gone from AGC to AAC, which means that transfer RNA is going to bring a different amino acid to the ribosome. And you can see that different amino acid here. So all the other amino acids have stayed the same because of that one base pair substitution. But because of that one change, that leads to a potentially different polypeptide, one that may not have the same function as it's supposed to have. And that's called a missense mutation. And so the effect then will be dependent on how critical that particular amino acid is for the function of that polypeptide or that protein. Now, in the next example of base pair substitutions, we're talking about nonsense mutations. And nonsense mutations involve where one base pair is replaced to another, replaced by another. So very similar to what we just spoke about. However, this substitution ends up producing what's called a stop codon. And we'll look at what that means in a moment. And there are 3 different stop codons that exist, which I'm highlighting at the bottom of the screen. And what this ends up doing when there's a nonsense mutation is the stop codon will actually produce a premature stop in the mRNA. In other words, they're going to terminate the translation of the protein or the polypeptide. And so our polypeptide or our protein will end up incomplete. So what does that mean? Well, once again, let's look at, we have normal DNA, normal mRNA copy of a gene, and then we have a polypeptide made up of the following amino acids. And so what we're going to look at here is CG, cytosine and guanine, in a normal DNA strand. When mRNA takes a copy, it has cytosine. These 3 nitrogenous bases, or this codon, is going to code for this amino acid in a normal situation. But in the case of a base pair substitution in a form of a nonsense mutation, we've substituted adenine thymine, which produces adenine in the mRNA strand. And what this does, of course, is change the sequence, and it now produces this UAA codon. This is one of 3 potential codons that, when present, can put a stop in the amino acid or in the polypeptide chain. And so instead of continuing on and with more protein, with additional amino acids forming a polypeptide, we end up with a premature stop. So ultimately, the protein is incomplete. Frameshift mutations are another type of mutation, and they involve insertions and deletions. Frameshift mutations are a type of base pair substitution, or rather, they're not a base pair substitution, they're a type of base pair insertion or base pair deletion. So this really involves then inserting or deleting one or more base pairs into the DNA molecule rather than substituting it. So you end up with a gain or a loss. And so these mutations will change the entire reading frame of the DNA sequence because this deletion or insertion is not a multiple of 3 base pairs. So it ends up causing a shift, and we'll look at that in a moment. And so what it does compared to our other examples is the amino acid sequence is going to be totally altered downstream from that point of the insertion or the deletion. So let's look at an example, and then I have another example on the next slide that should be helpful as well. So this is an example of an insertion, and you can see that if you compare to the normal DNA strand here, you can see that cytosine and guanine were inserted. So not substituting for another, it's not like one base pair is removed and this one is added in its place. It's completely just inserting itself, and it's inserted itself in between here. And so what this will end up doing, very similar to our other examples, is it causes a shift. It changes the sequence of amino acids afterwards. So you can compare then to here and to here. We have different amino acids, but not just one, but all amino acids downstream are going to be affected. So let's look at, and the same would apply with a deletion, but we'll look at an example on this slide so that it hopefully makes more sense. In the case of a frameshift deletion, you can see here that thymine and adenine, this base pair, has been removed. And when it's removed, everything is going to shift downstream. So if you looked up here, this would have been a codon, this would have been a codon, all coding for a specific amino acid, but now it's shifted where this is our new codon, this is our new codon, and so on and so forth. And then the same applies with insertion, which is an example we gave on the last page, where we're just inserting guanine and cytosine, again, shifting the entire amino acid sequence downstream. So this can have grand effects. But in simple form, if you think about if you're reading a sentence and I removed or deleted words, or maybe I inserted new words, it's going to change the nature of the sentence that you're reading and what it really means. So let's look at another example of base pair substitution. So we're going back to the first type of mutation we were referring to, where we could have a silent or missense or nonsense mutation. Here's a list of all of the different amino acids and then their 3-letter code. And you don't, of course, need to know these, but this group of 20 amino acids are going to, in different orders, create different polypeptides and different proteins. So if you're looking at this normal DNA sequence here, here's the normal mRNA sequence. And it's going to form a normal polypeptide. So you can see this codon is going to code for valine, this codon, and so on and so forth. And then it produces this beautiful polypeptide chain that was originally designed by the DNA within our nucleus. But if we look at another example, in a silent mutation, we have, in this mutant DNA, compared to normal, we've had the insertion, or substitution rather, of adenine and thymine. And so you can see the change here, substituted for guanine and cytosine. And so the mRNA copy then is going to consist of adenine rather than guanine. And so if these three, let's erase that so it's clearer, if these three code for leucine, with our base pair substitution, now we have these three. So we've gone from UUG coding for leucine to UUA, but look at that. That codon still codes for the same amino acid. And this is where the redundancy of our genetic code allows for certain things like this to happen without actually creating an obvious effect. So it's considered a silent mutation. So we did not change. You can see normal polypeptide. We did not change the polypeptide and therefore the protein. Looking at the next example of sickle cell mutation. We'll talk about sickle cell at a different point, but just to show you an example, so here's our mutant DNA compared to normal DNA, and we have mutant RNA because of this substitution here, which produced uracil as part of the mRNA strand. So we have the codon of GUA, which codes for valine. But what should have happened is adenine and thymine, copy being A, we have GAA, which codes for glutamate. And so you can see that there's been a complete change in the amino acid, and just this one change has had a great effect in creating what's called sickle cell mutation. So let's now speak about chromosomal aberrations or alterations. An aberration is defined as, or chromosomal aberration is defined as any change in the normal structure or number of chromosomes. And this can often result in physical or mental abnormalities. A few reminders of important terms for you about chromosomes. Human cells can be categorized as somatic cells and gametes. Somatic cells include all cells except your gametes. So the gametes, remember, are your sperm and egg cells. And each somatic cell has 46 chromosomes, or we can say 23 pairs. And we call these diploid cells. And they have the term euploid, and that's referring to them being in their normal state with their normal number. Now, each pair we know is donated, one by the mother and one by the father. We know that new cells are formed by the process of mitosis when it comes to somatic cells. Now, a somatic cell that does not contain the normal number of chromosomes, so 46, so if it has either less than or greater than 46 chromosomes, it's considered not euploid, which is normal, but aneuploid. We'll talk about those examples soon. Gametes, which we know are sex cells, are haploid. They're also euploid in their normal number. And we know that one member of each chromosome pair is going to be, so if it's the egg cell, it's going to be only the maternal DNA. If it's the sperm cell, it's only going to be the paternal DNA. But if a gamete cell contains less than or greater than 23 chromosomes in it, so an egg cell or sperm cell, it's considered not euploid, but aneuploid. So a reminder that this image here is showing you a karyotype, and I want you to remember that the first, let's get a pen, the first 22, so all of these here, are autosomes, autosomal chromosomes. And they're present in both males and females. And then this final pair here is the sex chromosomes, which are going to vary depending on if you're male or female. So XX for female, XY for male. And all of these that I circled, the first 22, are considered homologous autosomes. Each pair is identical in terms of the information that they carry. Now, this sex chromosome is only going to be homologous if it's XX, carrying the same information. It's not considered homologous then if it's XY, because the X and Y chromosomes contain different information. Now, everything I just said is summarized on this page, but we're going to now get into details as to why all of this information is important. So I want you to remember that a euploid cell is normal. So why is it important to look at differences in chromosomal number and structure? Well, chromosomal abnormalities are the leading cause of intellectual disabilities, as well as spontaneous miscarriages, which usually occur before the 20th week of pregnancy. They're common as well. They occur in about 50% of early spontaneous abortions and in about 1 of 150 live births. Two different types of chromosomal aberrations exist. We have numerical and structural. Starting with numerical, when a euploid cell, so a normal cell, is not a normal cell anymore and it contains more than the normal number of chromosomes, it is said to be, instead of euploid, it's said to be polyploid or polyploidy. And some examples, if you have a zygote that has 3 copies of a chromosome rather than the usual 2, then this is a form of polyploidy called triploidy. So 23 is the normal, multiplied by 3 of each instead of 2 of each, and that gives us 69. And nearly all triploid fetuses are spontaneously aborted or miscarried or occasionally a stillborn. Tetraploidy is a condition where a euploid cell instead has a total of 92 chromosomes, so referring to the 4 here, 4 times the normal number of chromosomes. We also have, instead of 2, we also have pentaploidy, so 5 times the normal number of chromosomes, and hexaploidy is 6 times. Triploidy and tetraploidy are the most common of all of these, triploidy and tetraploidy. They account for about 10% of known miscarriages, these two numerical chromosomal abnormalities. Now, a cell that has an abnormal number of a single chromosome. So going back here, remember, this condition of polyploidy is affecting all of the chromosomes. So they have extras of all of the chromosomes. So not just 1 or 2, but all of them. Now, in the case of aneuploidy, this is where there's an abnormal number of a single chromosome. So in the case of aneuploidy, it's important to consider the fact that you could have an extra or you could have one less. And the general rule of thumb is loss of a chromosome has more of a serious effect than duplication or having an extra chromosome. The first example is monosomy. Autosomal monosomy, so remember affecting the first 22 chromosomes, or one of the first 22 chromosomes. This is oftentimes lethal. And so these individuals, mono refers to one of their chromosomes is missing. So they might be missing this one, or they might be missing this one. This is usually lethal. Monosomy of the sex chromosomes. If someone just has a Y, they won't survive. We know how little that Y is and how little information it does contain, so these individuals will not survive. But monosomy of X can survive. So if you have a female that only has one X chromosome, monosomy of the X chromosome, she can survive. Trisomy, tri refers to 3 of the autosomes can exist, and when you have trisomy of 13, so 3 of 13, or if you have 3 of 18, or 3 of 21, these cases can survive. These are known chromosomal abnormalities of a euploid cell that can, or an aneuploid cell that can survive. Trisomy of the sex chromosomes. So if you have a female, let's say that has 3 X's, and this is the only example I'm giving you, oftentimes this extra X is inactive. So it ends up not being too much of a problem, and these individuals can survive. Now, structural problems also exist and they vary in type. We'll talk about deletions, duplications, inversions, and translocations coming up. So let's first look at an example of aneuploidy. This is an example of trisomy. in particular, trisomy of the 21st chromosome. So where we have, if you look around, you can see we have 2 of every chromosome in this karyotype, but here we have 3 of the 21st chromosome. And you would know then that this is Down syndrome because you've heard of trisomy of the 21st chromosome, I'm sure. This comes along with a lot of issues such as intellectual disability. There's an increased risk that they're going to have problems with their heart from birth. Congenital is from birth. Immunosuppression is also common, so they're more likely to develop conditions, respiratory infections, for instance. They're also more likely to develop cancer of the blood, so leukemia. There's also an increased risk of Alzheimer's-like or Alzheimer's disease-like symptoms after the age of 40. So if these individuals live until 40 or later, they start showing Alzheimer-like symptoms. 97% of Down syndrome is caused by what's called non-disjunction. So what does that mean? Well, remember thinking back a year ago when you spoke about how chromosomes can, when DNA replicates itself, and we have these chromatids or sister chromatids that are attached by a centromere during cell division, in this case meiosis 1 and 2, these should be separated and pulled apart. But sometimes they're not. Sometimes these sister chromatids don't undergo disjunction, which is normal. And if they don't undergo disjunction, what ends up happening is instead of, and this is just a very, very simple example, instead of one of these going here and one of these going here to a new cell, what ends up happening is this entire thing ends up going into one cell, which leaves nothing of that chromosome in the other cell. So what you end up having, let's say this is an egg cell, is that a sperm cell that unites with this one is going to produce 3, because it's going to give its 1 copy. There's going to be 3 in that final zygote of that particular chromosome. And then if it's this one that happens to unite with a sperm cell, what's going to happen? It's only going to have 1 in it. And so you'll end up with cases of monosomy or trisomy, depending on which egg is fertilized. So this is more commonly going to occur in females. And if we look at this graph here, you can see that this is maternal age. You can see that the risk of Down syndrome or cases like this non-disjunction, it goes up with maternal age. So chromosomal abnormalities are more common as a female ages, and you can see that there's quite a spike after the age of 35. Male sperm they don't, because they're produced all the time, ongoing, they don't undergo these same issues as frequently as females, whereas a female, her eggs are really as old as she is, so 35-year-old eggs. So when they start to age, they don't necessarily do what they're supposed to do all of the time. And the last thing I wanted to mention about Down syndrome is the life expectancy is 60 at most, provided that they survive beyond 10 years. If a lot do not survive past the age of 10. They also have a low IQ, and this constitutes their intellectual disability. In addition to the loss or gain of a whole chromosome, parts of a chromosome can be lost or duplicated as gametes are formed. And so ultimately this can change the arrangement of genes on a chromosome, so altering them. Let's start with deletions. So again, we're talking about chromosomal structural alterations. We just spoke about functional earlier. With deletion, some part of a chromosome can be broken off or lost, essentially deleted. So in this example here, if this is normal, in a structural alteration where deletion occurs, this part here ends up being broken off and deleted. And then what we're left with, this combines over here, is this here. So we've lost genetic information. This is considered unbalanced because of this loss. Similarly, you can gain parts of a chromosome. So for instance, if this here is duplicated, look how much extra information. And the same thing applies with when I spoke about how you're better off having more of a chromosome than less in terms of number. Same thing with structure. It's better to have more of or extra of than loss. And so this duplication is considered a gain, but of course it's still unbalanced. Now we have two other examples, inversion and reciprocal translation. With inversion, this occurs when there are two breaks that take place on a chromosome, followed by the reinsertion of that broken fragment at its original site, but in inverted order. So let's show you one here so that makes a little bit more sense. What we end up with is, with inversion, you can see B and D in this case are considered broken. So B breaks, D breaks. Then when they break off, well then they end up reinserting themselves, but in the opposite order. Now, unlike deletions or duplications as we spoke about earlier, there's no loss in this case and there's no gain of genetic material. So inversions are considered a balanced alteration of a chromosomal structure. And oftentimes they won't have any major or at least apparent physical effects. However, some genes can actually be influenced by their neighboring genes. In other words, if B, let's erase this, if B has the ability to be influenced by A or C, then it becomes a problem if B is now in a different place. And this is called position effect, which is just a change in a gene's expression caused by its position because of its influence of neighboring genes. And so sometimes this can result in obvious defects in this individual. Now, inversions can cause serious problems in offspring of individuals that carry the inversion. So for instance, if somebody carries this, you may not even know it. But then when they have a child, that child is now at risk of duplications and deletions because they inherited the inverted gene. Translocations. So this is reciprocal translocations. I'm going to first tell you what translocations are. A translocation, so this is different than reciprocal translocation. A translocation is the interchange of genetic material between non-homologous chromosomes. In other words, one chromosome can be broken, let's say chromosome 1, and it ends up becoming a part of, let's say, chromosome 8. Those are non-homologous chromosomes. They contain different information. Now, reciprocal translocation, as is the example listed here, this occurs when breaks take place in two different chromosomes, these homologous chromosomes, and the material is exchanged. So translocation refers to, think of it as moving from one area to another, but with reciprocal translocation, both chromosomes exchange their genetic material. So let's say, for example, we have this chromosome here and this chromosome here. If part of this chromosome breaks off and ends up joining in with this chromosome, that's called translocation. But in another example, let's say this part breaks off and this part breaks off. Let's say this is chromosome 1 and chromosome 7. Then this part of the chromosome that breaks off joins chromosome 1, and this part of the chromosome that broke off chromosome 1 is going to join chromosome 7. And so that's called reciprocal translocation. And you can see on this next slide what that looks like. So this is Philadelphia chromosome is an example of reciprocal translocation. And what you can see happening in this image here is that we've had breaks of a chromosome. So if this is normal here, you can see that there's been a break and an exchange. And this in particular is affecting chromosome 9 and 22. So two non-homologous chromosomes, they break off a piece of their chromosome and then it migrates to the other chromosome and kind of fits in its place even though it's not supposed to be there. And so this will end up with, you'll end up with this abnormal gene. And so this abnormal gene product ends up having the effect of increasing cell proliferation. And so Philadelphia chromosome is seen in leukemia, in most cases of chronic myelocytic leukemia, but also seen in other types of leukemia as well. So you can see its clinical effect. So we're talking about all of these genetic diseases, and there's the thought that, well, can we know early on? Well, people are starting to do genetic testing now even before they have kids, but if someone is pregnant, early on in the pregnancy, genetic testing can be done, so a prenatal diagnosis to see if there are any genetic abnormalities or chromosomal abnormalities, and at that point, a couple can decide if they want to carry on with the pregnancy, especially if there's a major chromosomal abnormality detected. And this child is very likely to not survive or not survive for very long if they are born. Then it gives parents an opportunity to make a very difficult decision. So there's two examples on here, amniocentesis and chorionic villus sampling, or CVS. With amniocentesis, as you can see in this image, a sample of the amniotic fluid is taken. And this contains cells from the baby, and these cells can be looked at to study the chromosomes, to see what type of issues might be present. Do they look normal? And this is one of the more common testing that's done, especially for women that either have genetic diseases that run in their family and they want to know if their child has inherited that, or if they're past a certain age where chromosomal abnormality odds increase, then they might choose to do amniocentesis. Another example is chorionic villus sampling, and this is where a sample of the chorionic villus is taken, if you think back to our discussions on embryology, and this structure contributes to the placenta, and it contains much more information, much more cells, but it's a trickier process. It's a lot riskier. You're more likely to cause problems, especially when you're inserting through the cervix. This can lead to premature labor, as just an example. So these are risky, but they do give us a lot of information as to whether or not that child has inherited a chromosomal abnormality, and these should be done early on. So let's talk about transmission of genetic diseases now and how they can be inherited. So let's go through some important definitions. First of all, the term genetically inherited, and they're grouped here where these are relevant to each other but different, and so on and so forth. Genetically inherited is something that you would inherit from, say, your mom or from your dad. It's some sort of gene alteration that can be transmitted to the next generation. Congenital, this is something that you're born with. So it could be something that's genetically inherited, but not necessarily. But it is present at birth. That's what congenital means. Now, acquired in utero, for example, back in the 60s, there was a drug called thalidomide that was used for things such as morning sickness, and it was found to cause major congenital malformation of the limbs of these babies. Another example, if a woman is exposed to a virus while they're pregnant, this can also cause genetic malformation. So the influence while in utero of some sort of outside factor that gets into the mother and impacts the development of her baby. And so this is much more common to happen early on in the pregnancy when development is happening. Autosomal, these diseases, you should know, these are going to affect chromosomes 1 through 22, whereas sex-linked diseases are going to affect chromosome X or Y. Recessive, this is a gene alteration in one allele that is clinically hidden. So these people are considered a carrier. So again, with recessive, Remember back to talking about genetics and how we have what's called a genotype, and this is just an example that I'm giving you. So this here is a genotype. And what you end up expressing ends up being your phenotype, right? The larger or capital letter represents a dominant gene, whereas the lowercase letter represents a recessive gene. The dominant gene dominates, whereas the recessive gene, there has to be two, or recessive allele, there has to be two present of these for it to actually take an effect. And we'll look at an example on the next slide. So if somebody does possess a recessive disease, but their healthy allele dominates and takes over, then this means that they are a carrier. So they carry that recessive allele and could pass it on, but it doesn't actually show up. And they may never know, if they don't have genetic testing done, they may never know that they're actually a carrier because it doesn't actually show up in their phenotype. And so I've already explained then what dominant is. So that's going to be representing in, whatever letters we're using, the larger letter that will dominate over the smaller letter. And so if you have a disease that's dominant and somebody has, say, this genotype, and the disease is dominant, then they will express it. Versus the disease being recessive, then they'll just be a carrier because the dominant gene will take over. So it really comes down to whether or not the disease is dominant or recessive, and we'll look at a good example coming up. The last two terms include penetrance and expressivity. Penetrance is the proportion of people that have the gene alteration and the clinical disease. So for example, if you have 100 people that have a specific gene alteration, and if all of them, so 100% of them, actually show the disease, so they have the gene alteration and they show the disease, then we would call this 100% penetrance. If you have 100 people that have the gene alteration, but only 50% of them show the disease, then you would call this then 50% penetrance. Lastly, expressivity. This is the proportion of people with a gene alteration and varied severity of the disease. So for example, if somebody has a gene alteration and the severity of it, so it's a very severe disease that they end up showing, then we would say that they have high expressivity. If somebody has a gene alteration, but the effect is the disease itself is very mild, then we could say that it has low expressivity. Some examples, for instance, are cleft lip and cleft palate. There's so many different degrees of severity. You can have someone just with cleft lip, with both cleft lip and cleft palate. You can have somebody with it unilateral or bilateral, mild, severe, etc. So this is a variance then in how it's expressed, and that's expressivity. Let's talk for a bit about how single gene diseases are inherited. And the two examples that we have up on the screen here are retinoblastoma and cystic fibrosis. Remember again that a capital letter is going to be referring to dominant and the lowercase letter is going to be referring to recessive. And each of these are alleles, and collectively they form a genotype. We have different types of genotypes. So for instance, homozygous dominant would be homozygous meaning they're the same, dominant meaning both letters are capital or dominant. Homozygous recessive, they're the same, meaning both are homozygous and both are recessive. And then heterozygous is going to be hetero meaning different, not the same, where you have one of each. So you can relate that then to our examples. Now in the top Punnett square, looking at retinoblastoma, you can see this is rare, less than 1 in 500 affected. But what I want you to be aware of is that this is a dominant genetic disorder. Autosomal, remember it's affecting the first 22 chromosomes, so it's not affecting the sex chromosomes, and it's dominant. So in this case, with our Punnett square, you can see that both parents are affected, and you can see the parents' genotypes listed here, as DB for both cases. That we know is heterozygous. If this is a dominant disease, then we know it's affecting the dominant allele. So this is the affected, and this is the affected. And if you inherit that affected capital D, or that allele, then you will have the disease. So if you have two affected parents, they know their genotype, and they say, we want to know what the odds are when we have children that they'll be normal or affected. And so you can create a Punnett square and show them once it's created. So DD gives us this, DD gives us this, and so on and so forth. What you can see is that anytime there's a capital D, these people are going to be affected because it's a dominant disease. So there is then a 3 out of 4 or a 75% chance that their offspring will inherit this disease of retinoblastoma, and only a 1 in 4 chance that they will be normal, because the smaller case D's in this case are representing the normal healthy allele, because this dominant disorder will affect the capital or the larger D. And so this is important information to give to parents then, so they have an idea of what their odds are. And also important to know is that it's not just these odds are not just applicable to their first child. The same odds are applicable to every time they have a child. Now let's look at a case of autosomal recessiveness, where both parents are carriers in this case. So what this means is that with cystic fibrosis, it's a recessive disease, meaning that it's affecting the lowercase allele. And so this is really what you would have to inherit, because it's recessive, in order to get the disease. Because let's say you inherited this. Well, if we know it's a recessive disease, it's affecting this lowercase b, or I should have done D, sorry, affecting this lowercase d, then we know that this dominant allele is healthy and it's going to dominate. So this person would be a carrier of it, but not actually display the disease. So if we look at the screen here in this example, the capital D's we know represent normal because it's a recessive disease. So this person is completely normal and not even a carrier, whereas this person would be a carrier but not actually display the disease. Same with this person here. But this is the situation where this would be displayed and this person would have cystic fibrosis. And so we could say then that there's a 3 out of 4 chance that they won't have cystic fibrosis. So 1, 2, 3. There's a 1 out of 4 chance that they will be perfectly healthy and not even a carrier. And there is also a 1 in 4 chance then, or 25% chance, that their offspring will inherit cystic fibrosis. And you can see that this is even more rare in 1 in 2,500. But carriers are common. This slide shows the continuum of the relative influence of gene alteration in human disease. On the right here, you can see that single gene diseases, such as cystic fibrosis and hemophilia A, are determined by genes or genetic factors. And therefore, they can be inherited. So genetic factors then play the largest role. And of course, then you have the ability to pass these on to offspring because of their genetic involvement. On the opposite end of this spectrum, we have environmental diseases. So things such as influenza and measles that are thought to be caused by our environment and our lifestyle, what we get exposed to, and do we make good choices to our health to keep our immunity high, etc. So they're thought to really have no genetic influence overall. And so they're called environmental diseases. Right in the middle of the spectrum, we have diabetes and heart disease. These are considered multifactorial diseases. In other words, there's environment and lifestyle that impacts the development of diabetes and heart attack or heart disease, and genetic factors can also play a role. And multifactorial disease are the most common, and so we're going to elaborate on that on our next slide, which is our last slide. Continuing on with multifactorial inheritance, so again, the middle of the spectrum on the last slide, these are caused by, as already mentioned, combinations of environment, lifestyle, and genetic factors. They are polygenic. In other words, there's not just one single gene that is essential for this. So for example, with type 2 diabetes, there are many different genes that could make somebody more prone to developing diabetes. And so the polygenic nature of these there's a polygenic nature then of these multifactorial diseases. There's so many different potential influences within our genes. Risk is also going to vary from one population to another. So just as an example, Canada versus Japan. Or there could be cultural, ethnic, environmental factors that differ amongst populations that can predispose somebody to this type of disease. And then some important examples listed here of multifactorial inheritance include cancer, include heart disease and cardiovascular disease as listed up on the screen, diabetes as well as obesity, Alzheimer's disease, and then congenital malformations as well, such as cleft lip and cleft palate, and congenital heart disease. So these are more common, the multifactorial inheritance patterns. And visit this website if you want to have your own genome made into a piece of art. I hope you enjoyed the lecture. Thanks for listening.